Welcome to the Broad Institute. We are delighted to have you as our guests to our first public lecture series, Midsummer Night Science. We hope to provide you with a better understanding of the human genome, our DNA, and how this genetic information is transforming our society. Here at the Broad, scientists seek to understand the very essence of our genes, written in the language of DNA, and to understand the basics of how they work and how they shape human evolution. These same scientists also use this information to understand how differences within our genes contribute to devastating diseases such as diabetes and cancer. Tonight, you will hear from Eric Lander, the founding director of the Broad. Eric will be weaving true tales of our biology that were written by evolution herself. We hope that you enjoy tonight's lecture, and we welcome your feedback. Please provide any comments on the cards that we provided and leave them with us tonight. And we do hope to see you again next week. For now, sit back and relax as Eric reads to us from the Genome Diaries.
Thanks, Michelle. Maybe you can bring up the PowerPoint stand. Well, welcome. It's great to have everybody here. The Broad is an unusual thing. It's a collaboration that stretches across our city, in fact, Cambridge and Boston, involving people from MIT and the Whitehead Institute, from Harvard in all its forms, the medical school, the Faculty of Arts and Sciences, the School of Public Health, the 17 teaching hospitals around Harvard. And the idea is to bring people together to work in a collaborative spirit to tackle some of the most important and challenging problems in biomedicine. That spirit of community, really trying to get the strength of this remarkable area that we live in, this greater Boston area, really is what the Broad is about. And in thinking about how we can best fulfill that mission, we also thought that reaching out beyond purely the scientific community, but to the broader community of Cambridge and Boston was really important because, well, this is the happening place in the world. Boston, broadly, Cambridge, Boston, this is Athens of classical Greece for our field. It's Florence of the Renaissance. It's just the most extraordinary time we are living through. And what's going on in this community is driving a revolution around the world. Of course, there are many people working on it in many cities around the world, but there is no greater density and no greater total push anywhere than there is within a radius of 10 miles around where we sit today. And so we think it's really important that we have a conversation not just amongst ourselves. We've been working on breaking down the silos between the different institutions in town, but breaking down the silos of academia in general, and taking a beautiful summer's evening and having a series of talks about what's really going on. I'm going to give a kickoff talk, and I want to describe this revolution that I'm talking about, this genomic revolution. I'll talk for a while, and then I'll be glad to take some questions. And then we do have more presentations from some very good speakers coming in future weeks, and you'll learn more in detail from them. Although Michelle said you should sit back and relax, you should in fact not. If you have a question, feel free to raise your hand and ask it or something. I'm going to try not to be burdened with jargon and all, but if there's something you want to find, just let me know, okay? So let's dive right in. It worked. Thank you, Stan. So I got to describe what it is that's going on right now, because it's cheap to talk about there's a revolution in science. You know, people always talk about revolutions in this, revolutions in this. I mean this in a very precise sense. Every scientific field, once in its history, undergoes a major transformation. That transformation is from having only a fuzzy description of its world to having a complete, a precise description of its terms of discussion. So for example, over there you've got a map of the Earth, circa 1350, state-of-the-art 1350 map of the Earth. You can see down here there's some Frigida, some Europa over there. Yeah, it's not bad, but I wouldn't sail west based on this map here, right? But, you know, now, of course, we have this picture of the Earth. We have absolute detailed picture. We have Landsat imagery. We have GPS coordinates anywhere on this globe. There are no more uncharted desert islands. If you sail west, you know exactly what you're going to hit. You today cannot imagine living in a world like this, what it was like to live in the world on the left there. There we go, to live in that world there. Your worldview is so totally changed by having the complete description that you can't get it out of your head anymore. That's what I mean about a change of worldview by having the big picture. This too, chemistry. It wasn't so long ago that chemistry was fundamentally alchemy. People going out, finding matter out in the universe, finding cool properties, you mix that with that, something happens, it blows up or whatever. And it seemed like an infinite, unbounded frontier there. But of course, along comes Mendeleev in 1869 with this periodic table and realizes that all of matter can be described in terms of a finite set of building blocks, about 100 elements. When Mendeleev made this table, he didn't have all the elements filled in. There were still about 20, 30 gaps. They spent 2 or 3 decades filling in all these gaps. But by the time they were done, these were the finite terms of discussion of which all chemistry had to take place. A student of chemistry in 1920 would not be able to imagine what it was like to be a chemist of 1880 or 1860 because you had this periodic table so firmly in your head. You knew these were the terms of discussion. That's what's going on right now in biology. In biology, we have had a world where it's infinite, unruly, unbounded. You want to go find the cause for cancer, people would go off searching for a new gene for this and a new gene for that or a new protein. And you could kind of always count on the fact that there would be some new undescribed gene or protein or something out there. And it would just go on forever. But what we're going through right now is a fascinating period over the past decade or so and into this next decade or so where we're getting our complete Landsat description of biology, our complete periodic table of biology, a complete description of what biomedicine has as its building blocks. And don't get me wrong, knowing the building blocks doesn't mean you know everything. Like knowing the elements of the periodic table doesn't mean you know everything about chemistry. But it is the essential terms of discussion of the field. And so in biology, one of the key events in that has been the sequencing of the entire human genome. We have a finite instruction book, 3 billion letters. That's it. We now know those 3 billion letters, give or take. And we need to understand what they are, so we need not just the 3 billion letters themselves, but we need to know everything that's encoded in that book. We need to know how much it varies between any 2 individuals, which particular genes are read out at what times in development and all that. But this is all a totally doable program. And the world of biology is saying, why can't we get all of that information, all the foundational information from medicine, collected in the course of just a couple of decades, get it all freely available on the web, and use it to drive a revolution by putting it in the hands of tens of thousands of young scientists. That's what this genomic revolution is about, and the worldview will be as fundamentally changed as it was for the other things I've described. The student in biology in the year 2020 will not be able to imagine what it was like in the 1970s, the romantic and barbaric world of the 1970s where biologists would go off into the jungle with their machetes, slashing, looking for a gene. Sometimes they would come back triumphant, gene in hand. Sometimes they'd never be heard from again. You know, this kind of very romantic picture of biology is being replaced by a systematic approach. Now, why should we care about this systematic approach? And what's this really, how does this all relate? Well, let me start by giving you a bit of history here. The idea that biology is fundamentally about information, that information at least lies at the heart of biology, is an old idea. It goes back to one of my great heroes, Gregor Mendel. Mendel in 1865 wrote this beautiful paper about the transmission of genetic traits in peas and was able to infer from the beautiful transmission patterns that there must exist genes that control them. It's an absolutely gorgeous paper. Needless to say, it sunk like a stone, totally ignored at its time. Nobody read it because it was so far ahead of its time. Mendel went into administration. It was pretty much the last thing you hear from him. Became the abbot of the monastery. But the idea was picked up again It was picked up simultaneously by 3 different groups around the world in January of 1900, right at the opening bell of the 20th century. 3 different groups rediscovered Mendel's laws of inheritance. And I like to read the whole 20th century as the development of that idea as biology as information. The first quarter of the 20th century to figuring out that the information resided in these pudgy little structures called the chromosomes. The next quarter of the 20th century being devoted to figuring out that the molecular basis of the chromosomal information was the DNA double helix. It's Crick and Watson there standing by a DNA double helix. And the next quarter of the 20th century devoted to figuring out how that information is read out from the DNA double helix, how it's copied into a molecule called RNA, a messenger, how it's translated from that RNA into the proteins according to a genetic code, and not just to working that bit out, but to working out the tools of recombinant DNA that made it possible for us to copy and sequence and read out that information. So that brought us 3/4 of the way through the 20th century with the ability for the first time to begin to read this genetic information. Now, first, individual genes, then sets of a couple genes, then the entire genomes of little organisms, medium-sized organisms, and then finally, in a wonderful set of bookends to the 20th century, the reading out of the nearly complete genetic information of the human being in the closing weeks of the 20th century. Going from the rediscovery of Mendel's laws in 1900 to the reading out of the complete genetic information of the human being in the year 2000. It's not bad as centuries go. Think what could happen in the next century if that same proportional progress could be sustained. So what it does is it brings us to a picture. I like to think about biology as having a vast library of information just ready to be read. There's a book on the shelf corresponding to every species on this planet. Actually, to every individual within each species. Not just that, every tissue within each individual, within each species in this planet, containing the information about its DNA sequence, its RNA expression of the genes, and the proteins that are there. And in principle, we can go to the shelf and pull down any of those books and read it. Now, the first couple books were a little pricey. The Human Genome Project, spent a couple $2 billion reading that book. But it's gotten cheaper and cheaper and cheaper and cheaper to do this. So that I urge young students today to imagine that they have the library card. They have access to anything in there. And if it matters enough, they can get it. And you might as well plan your experiments assuming that you could compare 20 different mammals if you wanted to, 200 different tumors if you want to, that all the information is there for the taking. Now, why is this powerful? The notion that this information could be useful for medicine. A nice example of that, 1980, here at MIT, in fact, David Botstein made a very simple and very powerful observation about how to use genetic information. I'll use it as an illustration of the power of being able to take generic approaches to things. Here's a family in which dad has a dominantly inherited disease. Boys are squares, girls are circles, he passes it on to 4 of his 8 children, the ones who are colored in here red. 4 of the children did not get the disease. Now, somewhere in dad's DNA, there was a spelling difference. On the chromosome dad got from his dad, there was an A at one position of the genome. Genome's 3 billion letters long. I left out most of the letters for you here. I just have one of them. And it's an A on the chromosome he got from dad and a C on the chromosome he got from mom. Notice that he passed on the C to all the kids who got the disease. And he passed on his A to all the kids who didn't get the disease. Mom, by the way, passed on A to everybody. I've contrived the example to keep it simple. So I got a perfect correlation between the passing on of this letter C and getting the disease. Does that mean that that letter C causes the disease? Well, no, it's only 8 kids. But what if I saw it in enough families? Well, after a while, it would become statistically pretty, overwhelming. And I wouldn't conclude it was the cause of the disease, but what I could conclude is that that spelling difference must be very close by on the chromosome to whatever the cause of the disease is, because it's always co-transmitted along in families. Now notice, I didn't tell you what the disease was. I didn't tell you if it was a brain degeneration, a bowel disorder, or a heart problem, because it didn't matter. It's an utterly, totally, completely generic strategy for mapping where the gene that causes the disease is without even knowing what it is, just by the correlation of the inheritance in a family. Now this was exciting because this could be applied to cystic fibrosis and Huntington's disease and neuronal polysaccharide lipofuscinosis and all these things because it was a generic approach, very powerful information-based approach. So in fact, they did this for cystic fibrosis. I remember in 1985, the day I walked into Building 56 over there, and David Botstein closed the door and he said, "They mapped cystic fibrosis. It's on chromosome 7. And because it hadn't been published yet, it was really exciting. And what they had was a genetic marker that showed 99% correlated inheritance with cystic fibrosis. Actually, the day he whispered to me, it wasn't that close, but within a few months, they got to 99%. Only 1% of the time did the genetic marker recombine genetically away from the disease gene. All they had to do was go from the nearby genetic marker to the disease gene itself. Problem is 1% genetic recombination corresponds to 1 million bases of DNA. So another way to put it is there were still 1 million bases of DNA away. So they had a genetic marker over here. It was sort of near cystic fibrosis. And then, not to get into the technical details, they took this piece of DNA and used it as a radioactive probe, washing it over other pieces of DNA to see what it would stick to, which would be the next piece. Took that as a radioactive probe, washed it over the pieces of DNA to see what it would stick to. That's the next piece, and the next piece, and the next piece, and the next piece. Every one of these steps takes a month and a half to do. And when you're all done, 5 years, more than 100 people, $40-odd million worth of effort, they got the cystic fibrosis gene. And it looks like this. Really boring. Lots of A's, T's, C's, and G's are the sequence of this gene. But I call your attention right there to those 3 letters, CTT. They're missing in the vast majority of people who have cystic fibrosis. That's the cause, the primary cause of cystic fibrosis. If in this room here, if everybody on the way out the door were to spit in a test tube, separate test tubes, please, we just take it upstairs to the 5th floor, we could run a quick genetic test and let you know in a couple hours who's a carrier for cystic fibrosis and who's not. In this room, there are 11 carriers for cystic fibrosis, approximately. And we could let you know who you were. So right off the bat, you get genetic diagnostics out of this. But not just that, you can find out what this protein does. You can take the sequence of that protein that you've sequenced there, chuck it in the computer and say, "Hey, computer, ever seen a protein, a gene that looks like this?" And the computer will respond, "Oh yeah, this looks just like 25 other genes that sit at the surface of a cell and transport ions back and forth. Congratulations, you found an ion transporter." Now notice, in the old days, when you found some new gene, you could pretty well be sure that you'd spend a decade working out what in the world it does. But here, all you had to do was put it in the computer, press return, it would compare it to everything else and hand you a hypothesis on a silver platter. Your gene likely is a transporter. And it turned out to be a transporter. And it only took a couple of months to confirm that, et cetera. Well, this was incredibly powerful. The idea that we didn't have to know what the gene was in advance. We could work from where it was to what it was. We could get its sequence. We could do diagnostics. We could figure out its function, all from that information. That's great. But what's the problem? The problem was it took 5 years, all these 100 people, tens and tens of millions of dollars. What smart young person would want to sign up to be an ant in that army, right? Science progresses by smart, young, ambitious people wanting to change the world. They don't want to be cogs in some big machine. They want to make a difference themselves. So we had a mismatch here. To solve these problems, you had to put together armies. What you wanted to do was liberate really smart young minds. That was the purpose of the Human Genome Project. The purpose of the Human Genome Project was to make it possible so that one bright young student, maybe an undergraduate student, in somebody's lab could knock off a problem like that much more easily. Now that would require, for example, having a genetic map those spelling differences I showed you of A's and C's and T's and G's, you need a genetic map covering the whole genome. You would need a physical map. All those pieces of DNA, which you had to schlep along, a month and a half and a month and a half, would have to all be done in advance and on the shelf waiting for you. All the DNA sequence would have to be there, so you didn't have to go back to the lab and sequence it yourself. You could just double-click with your mouse, and up would pop the sequence, and there'd be a list of genes there, and all of this information would be freely and immediately available without restriction to anybody in academia or in industry, in a first world country, in a third world country. That was the dream of the Human Genome Project because it would dramatically accelerate medicine. What actually happened? It happened that way. It's quite amazing. The scientific community came together, starting in the late 1980s with various discussions, launching in 1990 a Human Genome Project to try to develop all those tools, genetic maps, physical maps, sequence maps, all that stuff. It required a major change to the way we do biology. For example, back in prehistoric times, about 1987 or something like that, when you were sequencing, you would do it standing behind a plastic shield with radioactive DNA, reaching around, pipetting with your hand into a little glass, anyway. This is how we sequence today. This is at the Broad Institute, just 2 blocks away where we have our sequencing center. There are all sorts of robotics moving around, no radioactive anything. The robotics can move around, and we process something like 250,000 samples per day to get DNA sequence. And we generate something like 60 billion letters of DNA sequence per year, just here at the Broad Institute. That's, by the way, 20 times the sequence of the human genome. Hopefully, if we play our cards right, that is technology changes that are just happening now, if you come back for a lecture in 2 years, that number might be about 100 times bigger, because I think in the next 2 years we'll see about a 100-fold increase in that capacity, which is really exciting. But anyway, it's not bad as it is. So all this gets churned out. We had to rethink how to do biology in a more industrial form. Notice, by the way, it's not lots of people running around, pipetting and all that. There are a lot of people involved, but they're involved in planning the experiments, doing preparation in advance, analyzing the data afterwards, and we've got robots here as our friends doing an awful lot of the drudge work. It also involved a collaboration involving centers around the world in 6 different countries: United States, United Kingdom, Germany, Japan, France, and China. 20 different centers, some small, some large. The largest of the centers of the Human Genome Project was actually here in Cambridge. It was our own center here that contributed something more than about 35% of the total human genome. But everybody signed up for a common effort in which we all would make our data freely available on the web. There was, I'll note, a private effort to also sequence the human genome and not make the data available. And when you run a public effort that's making the data freely available and a private effort that's not making the data freely available, well, anyway, it doesn't end up making sense to run the private effort anymore because the public data are freely available on the web. And that was, again, how it sort of shook out, that it was not a great business plan to try to do secretly what, in fact, was being done publicly. But it was all good, and the human genome got itself sequenced, and that's us. In February of 2001, a draft sequence of the human genome was reported by this international consortium. In addition, the private group reported their sequence in February of 2001. It was not perfect by any means. It had lots of little gaps and imperfections. About 90% of it was there, 10% was missing, there were errors, et cetera. But it was already 90% of the whole human genome. The international consortium stayed with the program, and over the course of the next couple of years, produced a finished sequence of the human genome. Finished refers to the fact that almost all of those little gaps have been closed and errors and all that. There remain 321 gaps in the sequence of the human genome today, which we don't know how to close. Yes?
By finished, you mean the genes were
No, I mean the letters were there and correct. So we want to get 3 billion letters in order with a very high accuracy. So we have a string of, it's really pretty boring, 3 billion letters in a row. There are 321 gaps. And we know that the accuracy is, there are some mistakes, but the rate of mistakes is about 1 mistake every 200,000 letters or so. That's the sequence of the human genome. It fits on your iPod. It actually doesn't take up much space on your iPod. So you can carry around the sequence of the human genome. That's it, but that actually happens to be enough to specify a human being. Yes?
whose genome?
Whose genome? Well, we took pieces of DNA. They don't vary a lot, and we'll come to it. How much do you think they vary between each people? 5%. 5%. Turns out you only vary from you by 0.1%. Only one letter in 1,000 differs between any two people. So to a first order approximation, it doesn't matter who we took, because one letter in 1,000 would be pretty minor, right? As it happens, we took several people. We got samples from people. We anonymized the DNA so we wouldn't know who they were, and sequenced patches from different people so it wouldn't be any one person. But the plurality of the sequence comes from a guy in Buffalo, turns out. Well, that's because the lab that was collecting the DNA was in Buffalo, put the advertisement in the newspaper in Buffalo, and so we know it was a guy in Buffalo, but we don't know who it was in Buffalo. But, so if you want, it's a guy. There's also a lady in Buffalo, too. Yes?
Why can't you said technologically you can't do 100%. What's the technological barrier from getting?
Well, there are these stretches of DNA that go for a very long time. It turns out to be really tough to read through them and count exactly how many A's there are. There are other things like that that have like 3 letters that go on and on and on. Basically, the middles of chromosomes have these incredibly long repeats that we don't have any. We can sequence them, but we can't patch them together because they're so repetitive. You don't quite know what goes where. There are also a few pieces of DNA that for some reason we can't clone. But anyway, they're now down to curiosities. There are 321 regions that have some curiosities. It looks like almost no genes live in those regions. So for all practical purposes, we have the human genome sequence. But being intellectually honest, I say, you know, there's actually 0.7% that's not there, but it doesn't stop anybody in biomedicine anymore, right? Yes, please.
Skipped over there was a period where I remember there was quite a race going on.
Oh yeah, that's what I was referring to, this private thing, yeah.
I just wonder if you were planning to say any more about that, or whether in retrospect it really doesn't matter, the differences between the two approaches.
Oh, fair. I mean, the issue is never about differences between approaches. The issue is about whether data was going to be made public or not. That was the key issue in it. The approaches, everybody ended up using hodgepodges of different approaches. It made good news stories to talk about this approach or that approach, but technically, in fact, everybody uses a little of that, little of that. The key thing was, were the data freely available or not? That was the heart of what the controversy was, although it got expressed in terms of our method's better than your method, we're faster than you are, stuff like that. You know, it was the usual boys behaving badly sort of thing, but it served the public in that it accelerated interest, it accelerated some funding for it, it got the thing done. But the thing that really mattered, and the place where the public project could never lose, was if the goal was to get the information into the public domain, we couldn't possibly lose, and that's what mattered most, and it did get into the public domain. As it happened, I'll digress, the private group then dropped out and didn't continue with the finishing because there was no money in it. And we sort of had said that at the beginning, that unless your goal was to build a solid foundation of medicine, quite independent of any profit you'd make on it, you were never going to finish the job right. And so I'd argue that was basically right. You have to have a motivation consistent with making a public good in order to succeed in producing the highest quality public good. All right, so it's a beautiful genome. This is like chromosome 11 here. You'll see some regions of some regions very chock-a-block full of genes. Then right over here, a region very poor in genes. Gene rich, gene poor, gene rich. These gene rich regions here correspond to regions with lots of G's and C's, and to regions that are light bands in the microscope. When you look at the chromosome in the microscope, the light bands tend to be gene rich, the dark bands tend to be gene poor. Do you know why that is? Me neither. Nobody does. That's what's so cool about it. Nobody has a clue. It's fascinating. If you get to look at something like the genome for the first time, you say, wow, there's this interesting correlation. I wonder why that is. That's what's so cool. You land on a new continent. You see all these fascinating formations. And the next generation of students comes along and say, well, how are you going to explain this? How are you going to explain that? That's what's so cool about doing this. One of the big surprises was the gene number. I teach freshman biology at MIT, and I had taught a generation of students that there were 100,000 genes in the human genome. That was the official number. It was in the textbooks for 2 decades. Turned out to be a back-of-the-envelope estimate that a good colleague of mine at Harvard had made. And you know, that was such a nice round number, it stuck in the textbooks. But when we actually sequenced the human genome, it turned out that our best analysis found only about 30,000 genes in the human genome. This was disturbing for multiple reasons. One, because we'd been telling the students it was 100,000. but also because the mustard weed, Arabidopsis thaliana, for example, has 27,000 genes. It makes you kind of feel a little queasy being so close in your gene count to a weed. It's gotten worse, though, because it turns out that in that paper, that first paper in the human genome, when we analyzed it to count the human genes, we overestimated. Turned out that many things looked like genes, but they were really broken genes. We didn't have good enough sequence to tell. We now have a much more precise estimate of the human gene count, and it's 20,000 or so, just a little under 20,000. So we actually do have fewer genes than a weed. What can I say? Get used to it there. Actually, I can come up with a lot of good reasons why it doesn't matter. In fact, why it makes sense. You see, if you're a weed, you can't, and you're attacked, you can't run away. So a lot of genes are devoted to making secondary metabolites that make you unattractive to eat and other stuff like that. Whereas if you're a mammal, you can run away. But I think this is just sort of, rationalizing, actually. In any case, we shouldn't find our self-worth in our gene count, I think is what it says. But all of this is there. We sort of know our gene counts, et cetera. It's a beautiful genome. But of course, we want to know much more. And actually, I'll make a remark. 20,000 genes, it turns out we do, in fact, make special and good use of our genes. We can splice our genes in multiple ways, so a single gene can give rise to multiple proteins, 2 or 3 or 4 proteins. And a typical human gene gives rise to 3 or 4 different, maybe 3 different proteins on average. In addition, genes interact in all sorts of combinatorial ways. But anyway, the building blocks, there are only 20,000 genes. That's what it is. And that's very encouraging. It means it's a finite list. We can begin to understand what all of them do, et cetera. That's your human genome, your basic human genome. But let's go beyond that human genome. It's not enough to just have the letters A, T, C, G, et cetera, stretched out over 3 billion letters there. It's not enough just to have a list of the genes. We want to know what they're doing, how they're working. Well, the Genome Project had a pretty audacious goal. Find all the sequence. Check mark next to that. All, by the way, all is in italics. When I use all in italics, that's a technical term. It's a term of art here. I mean almost all, the vast majority of, 99.3%. Let's not fuss over the last 0.7%, right? I'll always use all that way. All means get the vast majority of it. So now here's what I think we got to do. The to-do list. We need to be able to read out this sequence and know all the functional elements that are encoded within it. The genes, but the regulatory controls, everything else that matters in that sequence. We need to be able to mark it up and say what matters. We need to know not just the guy from Buffalo and the lady from Buffalo, but coming back to your question, all the variation between people in the human population. It's only 0.1%, but it matters. Let's collect all that variation. We need to know all the signatures of cellular response. When you tickle a cell one way or another, when a cell gets cancer, what are those signatures? What are the ways the genes are read out? We need to know all the mutations that give rise to cancer. And we need to have ways to modulate, to turn up or down all the genes. That's a good to-do list, right? Fits on a yellow sticky. You put that up on the wall and you say each morning, let's do that. Well, that's actually what's going on. That's an awful lot of what goes on here at the Broad. And it's an awful lot of what excites this generation, is this is not a program for the next century. This is a program for at most the next decade. In fact, for many of these things here, tremendous progress has been made already. This is the second half of the Human Genome Project, so to speak. It's to get that full, rich annotation around the bare bones sequence so we can apply it to medicine. And that's what I want to now talk about. So finding all of the functional elements encoded in the human genome. How am I going to figure out what every nucleotide, or which nucleotides, which letters of DNA matter? I guess I could take a letter, change it, and see if it affects the person. Problems. A, completely unethical. B, totally impractical. So we can't do that. But that is in principle what you'd like to do. You'd like to change a letter and see if it matters. That actually is what evolution does. See, mutations happen at random. If they don't matter, they can be retained. But if the mutation affects a very important function, there'll be a selective disadvantage, and that will die out. So if we were to run a forward experiment, or actually let's go back 100 million years, look at an organism and see the mutations that pile up. Those things that can accumulate, can be mutated at a steady clip, don't really matter. Those things which never change must matter. So all we have to do is line up, say, the human and the mouse, or maybe a few other species, and see what is evolution kept and what is evolution allowed to drift. The stuff that matters should get kept. So sequence the mouse. So we've got to take all this DNA, figure out what's in there. Let's sequence the mouse. So we did. Together with two of our other friend centers at St. Louis and Cambridge, England, we produced a sequence of the mouse genome. Well, there it is, very nice genome. Line it up with the human genome. Turns out, if I take 1 million bases of human DNA, this is 1 million bases of human DNA. I left out the letters, by the way. This is supposed to represent 1 million bases of human DNA. For each piece of DNA in here, let me find the corresponding bit of the mouse genome that it matches to. The best matches to here. To this bit, the best matches here. The best matches here. Best matches here. In fact, this 1 million bases of human DNA matches up exceedingly well with this 1 million bases of mouse DNA. You know why? Because this descends from 1 million bases in our common ancestor. And you can quickly see it in the DNA, that there's a sequence that was there, and it descended along the human lineage, descended along the mouse lineage, and you can just go line up this 1 million of human, this 1 million of mouse, and they clearly match up exceedingly well. In fact, across the whole genome, you can see this bit here on mouse chromosome number 15 matches up to a stretch on chromosome 8. This bit here on chromosome 18 matches up to human chromosome 18, this bit, et cetera, et cetera. There's a whole lookup table. For any part of the mouse genome, you can get the corresponding part of the human genome. For any part of the human genome, the corresponding part of the mouse genome. There have been some breaks and rearrangements, but only about 330-odd breaks and rearrangements. Otherwise, you can match up those two genomes very closely. You know, when people argue about did evolution happen and all that, I mean, we can sit here with 2-3 billion letter sequences and trace out what went on when. And you imagine if I had this for the dog and the cat and the cow and the whatever, we can tell you, oh yeah, this letter changed after this, but before that, et cetera. You can do all that. It's not about fossils. It's actually about an extraordinarily rich amount of information that documents the precise changes that went on at different times. And we read all this out. So, I mean, to me, it's slightly surreal when people argue about did evolution happen or not. Because we actually are not just constructing that it happened, but the precise details of where the breaks were, what the nucleotide changes were, et cetera. So if I take sequence and I run it through the computer, I can now look for the stretches. I'm going to go back for a second. I want to take the sequences of a human stretch and a mouse stretch. I now want to look for the bits that have been exceedingly well preserved by evolution, that evolution hasn't allowed to change very much. So this is just a cartoon, but if I give you a stretch of letters here and a stretch of letters here, can you find any hidden message that's been well preserved in this? This is hidden, right? Computers are really good at doing that. So you can take human DNA, mouse DNA, run it through and say, find me all the places where there's like 8 letters in a row that are still identical or 20 letters in a row that are identical. And it'll match it up and find the bits that are very well conserved across the whole genome. Well, it turns out that when you do this, for a typical gene, don't worry about the details here. This is a gene we happen to like. It goes by some funky name, PPAR gamma. Don't worry about it. Some bits of the gene are very well preserved by evolution, and they encode the protein. And that should be no surprise, because everybody knows the protein matters a lot. But it turned out that there were also a lot of parts of the gene that were very well conserved by evolution and do not encode the protein. Do you know what they are? Me neither. That was what was so cool. The official textbook picture said that what mattered was the protein and maybe a little bit of regulatory stuff at the beginning. Turned out to be exactly the opposite. Evolution has been lovingly conserving 5% of the human DNA, 5% of your genome has been very well preserved by evolution, not allowed to drift very much. only 1.5% of that encodes the protein. The other 3.5% encodes we don't know what. But it clearly matters because over 100 million years, evolution hasn't let it change that much. Students love this because it means the previous generation completely missed the boat on this point. That there was a lot of stuff that matters in the human genome. And now we're beginning to figure out it's regulatory stuff that tells you when to express this gene in the liver, in the heart, at this time in development, et cetera. It's RNA genes, it's structural things. We didn't have to know what it was. All we had to do was count on the fact that if it had a function, evolution wouldn't let it change very much. And we now have a markup. Now, of course, I'm cheating a little bit. Mouse and human alone are not enough to do that markup precisely. You can get pretty good. But if you want to get it precise, like this letter matters or these 4 letters matter or something, maybe you want the dog also. So we sequenced the dog. Maybe the armadillo. So that's been sequenced. Cat, yep. Elephant, yep. Actually, by today, 18 mammals have been sequenced. By the end of the year, it should be up to about 24 mammals will have been sequenced. The data are all freely available on the web. Come visit our website. You can look at analyses of this. And with that, we can see exactly what evolution's been preserving, what it's not been preserving, when certain changes happen, what changes happen. Just recently, we sequenced the chimp, and there are actually some very interesting genes that have had a lot of change just since our divergence since chimp, but otherwise are well conserved in other mammals. You can read out the whole storybook of evolution from this information. I can't say we understand all of it yet, but you can read out the story, and now students are pouring all over this saying, well, what does it mean that this bit is conserved, or that this bit changed in this lineage, et cetera, et cetera, et cetera. This is what I mean about the information revolution in biology. So anyway, you can get all that. Now, sometimes we can figure out what these things are. Here's a particular 401 bases or so that's been very well preserved, 84% identity between human and mouse. A friend of ours in California knocked out that particular segment. It affects the control of a whole bunch of genes in its neighborhood. That's a gene regulator. It's one down. There's about 500,000 such things to go. And students are all over this. They're obviously not going to do it one by one. They're thinking about ways to do this in high throughput. And people want, over the course of the next 5, 6, 7, 8 years, to get a pretty good accounting, and I think they will, of what all of this extra stuff that functions in the human genome does. All right, that's part 1. Check on that, all functional elements in the genome underway. All human genetic variation. Well, let's come back to your question. How much do any 2 people differ? Here's sequence. Any 2 people differ by that much. That's why we didn't care if it was the guy in Buffalo or lady from Miami, for that matter. That's 1 letter in 1,300, actually. I said 1 in 1,000 to be round, but it's actually about 1 in 1,300, a little less than 1 in 1,000. That's actually not a lot of difference between any 2 people. Any 2 of you are 99.9% identical. Actually, very little as species go. If I take any 2 chimpanzees and I compare them to each other, they differ on average by about twice as much as any 2 people. If I take any two orangutans from Southeast Asia and I compare them to each other, they differ by about 8 to 10 times as much as any 2 people. See, you think the orangutans all look the same. They think you all look the same, and they're right. Right? We have very little genetic variation as species go. Now, why is that? Because we all trace back to a small founding population. All humans trace back to Africa, some 3,000 or 4,000 generations ago, to a relatively small founding population with an effective size of about 10,000 individuals. There's a very simple back-of-the-envelope formula you can write in population genetics that says the amount of genetic variation that a population has is directly related to its size. We have exactly what you would expect for a population of about 10,000 individuals. But you say, wait, we're 6 billion people. But that's very recent. That's in the past, not very long at all. In fact, we walk around today with the genetic variation that we walked around with in Africa because there hasn't been enough time for a lot of new mutation. Again, you can do it back in the envelope. We know what the rate of mutation is. It's 2 times 10 to the minus 8th per generation. There hasn't been enough time for there to be very much mutation. Now you say, okay, I don't believe that. Do the following experiment. Take DNA from a person in Japan, a person in Africa, a person in Sweden, and compare them. Do you see the same genetic variations? The back of the envelope calculation says that 95% of the genetic variation that you'll see in Japan, you'll see in Sweden, you'll see in Africa. When you actually go do the experiment, that's exactly what you see. Most genetic variation is shared genetic variation amongst all populations in the world, and is the genetic variation ancestral to the whole human population. It's the variation we walked around with in Africa. Yes?
You were saying just before
Yeah.
about the bits. Is any bit in the sequence, does it have the same significance? I mean,
No, some bits matter tremendously. Some bits,
So
you
is-
change it and you have you're dead.
So what I'm wondering is there a point at which there's sort of a minimum threshold at which the length of the bit can be meaningfully matched or not matched? You see what I'm getting at?
I do, and that's exactly what I was sort of getting at before and figuring out which bits are functionally important and which aren't. There are some places where a single letter matters a lot. There are some places where 300 letters doesn't matter, they could come or go. But the way we test that is asking evolution. Does evolution allow it to change? And we can tell what bits matter by the fact that they don't change over evolutionary time very much. So it's a very uneven patchwork of what matters and what doesn't. So human genetic variation, in principle then, maybe since there's not so much genetic variation, we could collect, yes?
So along that same line, are there areas where there's a lot of genetic differences, and we just say, you know, it looks like nothing really is going on in these areas on the genome?
Interesting. There are a few areas where there are a lot of genetic differences, and a lot's going on. There are some areas like that affect your ability to deal with infectious disease, where in fact we have a lot of variation precisely so that no one bug can kill us all.
So I guess for the 3 billion letters,
Yep.
is kind of the idea that every letter is important?
No,
or
no, only
a lot of
90
letters don't really matter?
Well, like we figured out with evolution. Evolution told us 5% really matter and 95% don't matter a lot. So that's right. So you want to know which of these genetic differences matter, which don't. Let's take a specific example, okay? Let me pick two. Can I pick two letters for you here? On chromosome 19, here's a gene. It goes by the name apolipoprotein E. It's got a long DNA sequence, and I've left out everything except these two letters. Some of you have a T at this position and a T at this position. Some of you have a T and a C. Some of you have a C and a C. If you happen to have the CC on the chromosome you got from mom and the CC on the chromosome you got from dad, you have about a 70% risk of Alzheimer's disease in your life. And if on the way out the door you spit in a test tube, we could go up to the lab and leave a message on your answering machine tomorrow. No, we couldn't do that because it's unethical, right? To call you up and say you have a 70% risk of Alzheimer's disease. But technically it's not difficult, right? We could do that. And in this room, there are 7 of you who are CC homozygotes and have a 70% risk of Alzheimer's disease. I could do this on myself. I haven't because there's actually no treatment for it. There's nothing I could do about the action, and therefore all I would do is get depressed. So I'm being serious. This is a serious analysis of the problem. The minute there's a drug I could do to delay the onset of Alzheimer's disease, I'm going to go find out. So pharmaceutical companies now are using the fact that this gene is involved in Alzheimer's disease to go try to make drugs to slow down the process, et cetera, et cetera. Some of those letters matter a lot. But if I move like 12 letters over to the left, probably doesn't matter at all, that letter. The details matter, right? Spelling counts, right? We can give you more examples of this. And in fact, David Altshuler, in the last lecture of this series, is going to talk a lot more about human genetics. But there are, examples and examples and examples of gene differences that, here's a good one. In this room, there are 2 people who have a deletion in the gene for CCR5. You're immune to AIDS. Because it turns out AIDS needs that protein to infect you. 2 of you are resistant to AIDS because you have a deletion in that. Now, that's good for the 2 of you, but it's actually good for the other rest of you because knowing now that fact, we can now make a drug that tries to have the same effect by blocking that protein. That's why we want to know these things. Anyway, here's the real dream, and David will tell you a lot more about this later. Turns out there's only about 10, 12 million genetic variants at any appreciable frequency in the human population. Why don't we just get one very big Excel spreadsheet, write down all 10 or 12 million genetic variants across the top of the spreadsheet, and figure out which ones are enriched in diabetes, which ones in arthritis, which ones in stroke, asthma, et cetera, et cetera, et cetera. And just have one very big lookup table. Here was Alzheimer's, we had these two, but here's early heart attack, here's that. Human geneticists are saying, just like we got a sequence of the human genome, now let's get a complete database of all human genetic variation between people, all 10 or 12 million genetic variants, and let's go work this out. Well, that's a nutty idea, except that it's happening. Over the past not so many years, the database of human genetic variants has grown and grown and grown to the point where more than 8 million of the human genetic variants are already identified, freely available on the web. You can come to our website, look at that. And already the tools to be able to look at their correlation with disease are progressing. And you'll hear about that in the fourth lecture there. So that's the topic, the second thing, know all human genetic variation. In the course of this decade, we will have a comprehensive survey of all human genetic variation and greater precision as to how it relates to the cause of disease. I'll take one more topic here, all signatures of cellular response. Another good application of the human genome. Here's two cancers. These are two leukemias. Two different patients came to the clinic with leukemia. Is there a difference between these two? Well, not by eye. No pathologist can distinguish these by eye. But it turns out that these are very different leukemias, and they need to be treated totally differently. But this took a tremendous amount of work, 40 years of work, starting with the work of this man here, Sidney Farber, the person after whom the Dana-Farber Cancer Institute is co-named. Sidney Farber, by looking closely at these leukemias, was able to tell certain very subtle differences in the nucleus of the cells, and then some funny enzyme differences, and some funny things on the surface, and then some stuff with the chromosome. Anyway, after 40 years of work, there's a rock-solid, really clean distinction between one leukemia called AML and another leukemia, ALL. And it's really important. First thing a pathologist has to do is figure out whether the kid who's presenting with this cancer has flavor A or flavor B in order to figure out how to treat. It's a triumph, except that it took 40 years. What about all the other cancers where there's some distinction we haven't discovered yet? Wouldn't it be possible to just kind of peer down into the DNA or the RNA or the protein and just automatically say, Zoya, of course, they're totally different cancers? Well, you can do that. Turns out that instead of looking at the DNA sequence, one approach is to look at the RNA, that is how the genes are being read out into the cell. You can take these things called gene chips, little slide of glass here. Every one of these squares has a piece of DNA on it corresponding to a particular gene. Gene 1, gene 2, gene 3, gene 4, et cetera. It's actually a little more complicated than that, but for all intents and purposes, there's a little thing corresponding to each gene. Take a tumor, grind it up, prepare its RNA messages read out from the genes, label it with some fluorescent something, squirt it onto the chip, swish it back and forth, stick it under a laser scanner, and read out every gene. Each gene, gene number 1 will stick to spot 1, gene 2 will stick to spot 2, gene 3 to spot 3, and if there's a lot of gene 3, you'll get a strong signal. When you do that, every tumor, here's a tumor, can be read out as a vector, a string of numbers. Here are genes running this way. This gene is high, red means high. These genes are highly expressed, these genes low expression. And I've left out a bunch of the 20,000 genes here. So every single tumor is a row in this spreadsheet. And these genes are high in ALL, these genes are high in AML. There's an obvious molecular difference. So I'm going to give you a new tumor here at the bottom. Patient comes in, we prepare it, we do a DNA chip. Is that AML or ALL? AML, good. Next. What's that one? AML, very good. Next. Congratulations, you're pathologists. Very good. Turns out the computer can do this with essentially perfect accuracy. The genetic difference that can be read out is sufficiently striking that it can be read by looking at its genomic readout. Now, that's great for AML and ALL. And the pathologists say, okay, well, it's great, but we figured that one out already, thank you. But what's been going on since then is people have been doing this now and showing that there are types of leukemias that we hadn't recognized. Turns out ALL itself comes in 2 flavors, type 1 and type 2 that we hadn't known about. Lymphomas, breast cancers, colon cancers, et cetera, et cetera, et cetera. One is able to take a totally generic approach. Oh, actually, now wait a second. I cheated for a second, didn't I? I gave you I gave you a whole bunch of known examples, and I had you compare to a known example. Sidney Farber didn't already know the difference between AML and ALL. So what he had was this problem. He had a whole bunch of cancers, and he had to say, were there two subsets there? So if I gave you this data, do you think you'd be able to recognize two subsets? Turns out that if your eye is good, you can kind of see stuff going on here. Now, could you really recognize this? For the computer aficionados, every tumor is a string of 20,000 numbers corresponding to the 20,000 genes. That's a point up in 20,000 dimensional space, whatever that is. What we're really asking, if every tumor is a point in 20,000 dimensional space, do they cluster in two corners of space? Well, whatever I just said, you can imagine you could write a computer program to figure that out, because computers don't mind thinking about 20,000 dimensional space. And when you do that, the computer takes the data without any knowledge of AML and ALL. You press return, and 3 seconds later it says, bingo, they sort out into two very distinct groups. So the truth is, you could do this without any advanced knowledge of AML and ALL. And that's what's going on now with these lymphomas and these colon cancer data. The data itself are sufficient to divide themselves, to give themselves their own proper names. Anyway, I could go on with this stuff, but you have a sense of what I'm talking about. Biology is turning into an information science. What started with Mendel and the ideas of some laws of inheritance is growing and growing and growing to saying we can get a complete readout of the human genome. We can get all the functional elements by comparison amongst organisms. We can find all the genetic variation between people and figure out which correlates with disease. We can find the different subtypes of tumors by looking at their informational readout of what genes are on and off. Notice, I haven't yet said on my to-do list, we're going to cure all disease. That's a different promise. I'm not promising that all disease gets cured. What I am promising, I'm just going to skip through because, what I am promising, and then I could tell you similar things for knowing all mechanisms of cancer and modulating all genes. There are ways to do all these things. What I'm saying is a set of tools are becoming available right now to let us being the world, us being this extraordinary next generation of young people coming into science, take on problems in a powerful way. You want to find the basis of cystic fibrosis? If you had to start that problem again today, be done in 2 weeks by one good student. I know that because I know other genetic diseases that have been knocked off by one good student in 2 weeks. Cancers, finding all the things that cause cancer, still a little hard. Come back in 5 years. I think we'll have a pretty complete classification of what are the mutations that give rise to cancers. We're going to move into a world where all of these extraordinary young people trying to tackle the important problems of the disease are going to do so with a set of tools, databases of information, ways to probe cells, DNA chip sequencing, et cetera, that are going to let them overwhelm problems. It does not guarantee that we will cure everything. But what I can guarantee you is we had no chance of trying to cure things without knowing this, without even being able to say there are two totally different types of leukemias here. There's no way you can approach a cure. Recently, people here figured out there's a subtype of lung cancer. It's 5% and it's very distinct. It responds differently to drugs. That's great, but what of all the other subtypes? This is the revolution we're going through right now. It's a revolution which by the time we're done, it really will be unimaginable how we ever sought to take on disease beforehand. It will be something which I feel confident saying 50 years from now will have an extraordinary impact on cancer, an extraordinary impact on most of human disease. But I'm very cautious about promising that's going to happen in 5 years. I'm not counting on this for me, but I sure am counting on this for my children's children. It may be that it happens faster than that. This may affect my children. I hope it will affect my children. It will in little ways already. Some drugs are happening, et cetera, but the full revolution, from the time people discovered the idea that infectious disease was caused by microorganisms to the time we really had effective antibiotics, 60, 70 years. That's shortened the span of human history. If we can, over this next half century, pull this full program off, the next decade of getting all this information, and a couple decades to come, turning this into effective therapeutics, we might reach mid-century where cancer is merely a chronic disease that's treatable and not a fatal thing. And I will not apologize if that's what happens, if it takes that sort of period. That's a period on which, although it may not be attractive to Wall Street to say I'm willing to invest, I'm certainly willing to invest for my children and my children's children. That's what's going on. It's a change that happens once in every field. It's a change that's happening right now in biology, in biomedicine. And it is an amazing time to be alive and be in science. Some of you are young people probably still thinking about what you might want to do with your careers. Come on in. It's just, the water's just great. For everybody else, at least know what's going on with this. There are many important questions right now about what we should be doing, what opportunities in research we should be taking, what funding we should be devoting to this. It's depressing to say that we're going to see this year the first federal budget which actually cuts money, decreases money for basic research and for medical research. I can't understand why at this point with this opportunity we would choose that as a priority, to start decreasing the budget for the National Institutes of Health. But it's a decision we make together as citizens, and it's all of our voices that matter. Anyway, it's an amazing time. I'm so glad that you would choose on a midsummer night to come out to the Broad and hear a bit about this. Thanks for joining us. Thanks for being part of the Broad community. And maybe I'll take a couple of minutes of questions. Thanks very much. Some questions. There's one and there's one. Yes?
Referring back to your earlier point of public versus private efforts in the sequencing of genome.
Yes.
The importance of making that, putting that in public domain. In that context, what's your opinion on the patenting of human genes and proteins that encode, regardless of whether any application is specified? Does that defeat part of your earlier purpose?
So this is a very important question, the patenting of human genes. So let me say for starters, patenting can be a very good thing. If somebody discovers a new drug that will cure a particular type of cancer, I'm all in favor of them having a patent over that particular molecule. Because if they don't have a patent over it, they won't go invest the $100 million it takes to run a clinical trial to prove it. Because if they didn't have a patent, somebody else would just swoop in and sell the molecule. So there's a reason why we as a society grant a monopoly over a drug. Because it makes people be willing to invest to do that. But it's a totally different question if I'm just running a sequencer, churning out data, and have made almost no contribution to improving the world by finding a bit of sequence, making some kind of a prophetic claim that I have not really done anything to realize, and saying, land grab, I want to claim this. You know, back in the Homestead Act in the 1800s, you didn't just get a piece of land by walking the boundaries. You got a piece of land by working the land for 7 years. It's the same sort of thing here. We make a social bargain. We're willing to give inventors monopolies over things if they've truly improved it and worked hard for it. So I'm not at all opposed to patents, but I am opposed to this idea of granting patents willy-nilly for what are not really substantive inventive acts that ever make a difference in therapy. Happily, many of those patents are not going to have much of an effect. Many of them won't have issued. But I am worried about further claims and things like that. There are nutty claims the Patent Office allows today. If you get one drug that works to cure something in a certain way, you can file a patent today that says, I now claim all possible drugs that work in this way to do something, without having even described what the drugs are. Some of those have issued. It's nuts. We're not getting our money's worth as a society in granting monopolies for that. So it's a technical and an economic question. What's the trade? What's the balance we're willing to make? The patent system is an important part of this, and I think it's been way out of whack. And we do have to make sure that we are getting our money's worth when we choose to give a patent, because otherwise we're going to disincent people from doing the hard work. Finding the genes, that is a public good. It's a public service. We get that information out there for everybody. Many other aspects of this, all human genetic variation, should be freely available on the web so people can use it. Many of these things, descriptions of what's wrong in cancers. Patents should start when we actually start helping people with therapeutics. A question here.
In young people coming along and being interested in this.
Young people is the fuel of progress.
And what I found very radical in what you said was that biology is becoming more of an information-based or special.
Science, yeah.
Science. How do you, if you wave your wand,
Yep.
how would you change how we bring along young people into the scientific field? It sounds to me like programming, you keep saying, and you can just go in and program that computer to do that.
Yeah.
It's
So-
just basic math?
No, it's not just basic math at all. It's a mix. You have to feel comfortable with math and computer science because you're going to be slinging around data.
And when do you think that should start?
And you also have to be comfortable with biology and medicine because it's about an organism, it's about a person, a human being. 20 years ago, 15 years ago, we talked about wet scientists and dry scientists because you either were trained in wet biology or you did some mathematics. The students who are coming along, the best ones today, are sort of comfortable with both. They're conversant. They grew up with computing. They grew up with software. They're also interested in the biology. They shouldn't even see it as a divide. When I say biology is an information-based science, I mean you care about the organism, and you see the organism as a beautiful organism. You see it as its tissues. You see it as its molecules, and you see it as its information. And you can go back and forth across all those levels of understanding.
So in our schools,
So what's,
there should be more convergence.
well, more convergence. You know what I think there should most importantly be?
There's a lot of studies out there that say kids who get, it's really by middle school that kids decide internally sort of how they're going to proceed.
There should be more excitement. In what I described to you, on several occasions I said, you know what this is? Well, that's news to me. I didn't know either. Students need to hear that. What they get in the textbooks is, here is the received wisdom. You know, you can open the book and it'll tell you how the plant works and the xylem and the phloem and the da-da-da-da-da. What's exciting right now is we're finding out all the stuff we didn't know. We're finding out that the last generation left an awful lot unaccounted for. We're finding out that this is the best time to come in because of the uncertainty, the excitement. It's terrible for students not to find that out till graduate school. To imagine that the world is all completely figured out and the goal is to memorize what's in the textbook. Some reasonable fraction of what we teach students should be about what we could have taught them 10 years ago and how much more we can teach them now and how much we still don't know but are likely to know in the next 10 years, and then how much we're going to know in their career as a scientist. Mostly what it's about is to recognize that it's fast-moving, it's change, it's exciting. And we somehow manage to wring that almost completely dry from the way we teach science at young ages. I want them to know it is a world of mysteries and a world of opportunities and possibilities, and the tools are here. And so when I give this talk, for example, I try to point out those sort of things. Let's get that to our students, because the rest, then they'll go pick up they'll pick up the computer science, they'll pick up the other things. They have to think it's worth it. They have to think they're going to matter. They have to think they are the generation that is going to lead us into this new frontier of biology, and they are the generation that are going to produce the therapies for our children and our children's children. Yes?
So, given what you just said about textbooks, and for a non-specialist who would want to understand this better, is there either a good book or a good article or articles that you can recommend that provide a good
Golly. There's a lot of good stuff nowadays being written. And I might turn to my colleagues, Finton and Michelle, and ask, maybe, as we run the Midsummer Nights lecture series, as we get to the end, we might put up at the end a suggested reading list of further reading for people here. Rather than I give you one or two off the top of my head, let's put up some good reading so when you go away in August, you can continue the spirit of the Midsummer Nights lectures. Question there and then there, yes. We'll take just a couple more, yeah.
Definition of a gene.
What's the definition of a gene? That is what is so wonderful about definitions. I was trained in mathematics. You make a definition, you use that definition. You can make another definition. Turns out there are about 12 possible ways I can use the word gene. I know which one I'm using at any given point. Mendel, who didn't actually use the word gene, but in any case, what he was talking about, the word gene was coined, I think, by Johansen in 1904 or something. What they were talking about was whatever it was that got mutated that caused the pea plant to do this or that. That's a definition of gene. The thing, the abstract thing that causes that. Another definition of gene could be when you started doing genetic mapping, you can map things by recombination. It's 1% away from here. It's a region of a chromosome that controls a trait. Another could be the specific DNA sequence. Another could be the specific DNA sequence plus the RNA that is read out and spliced up in a certain way. And the thing is, any of those, as long as we agree on our terms of discussion for a given conversation, we can use. So the problem is when the general public talks about gene and they try to make sense out of what scientists are saying, at any given point in a technical discussion by scientists, we know which precise meaning we mean. Now, what did I say when I said there were 20,000 genes? I meant there were 20,000 regions of the chromosome that encoded one or more overlapping RNA transcripts that were translated into a protein. I did not distinguish between different RNA transcripts at the same region, nor did I allow into my count RNA transcripts that were not transcribed into a protein, nor pieces of DNA that might have a function but not make an RNA. But that would be really cumbersome to say, so I just said 20,000 genes. Okay, but that's what I mean. You can use precision, or you can just sort of let it slide, and most of you were kind enough to let me let it slide. There was a question over here. Yes?
You were alluding to the rapidly accelerating speed and sequencing technology.
Yes, rapidly accelerating speed of sequencing.
When do you think will a whole genome sequencing be available at an affordable price as a routine diagnostic tool?
Whole genome sequencing available at an affordable price as a diagnostic tool. Well, it partly depends what's affordable. So there are new sequencing devices that are in principle capable of sequencing about 1 billion bases for about $3,000. You would have to sequence about 100x coverage. So currently, to do what you want to do, I could probably do it for about $300,000 or $400,000 today. It's not an affordable price, but you're asking when would it, now, of course, that new sequencer that I'm talking about, as of June 30th, like didn't exist in anybody's lab. But the first of those sequencers was in fact delivered last Sunday. We got it. And so we've been running it for the past week. So I don't actually have enough experience to say how well it's really working, but we got 2 runs off it so far. And it's still not at its full density and yada, yada. Anyway, I don't know, $300,000 or $400,000 to do a genome now, way too expensive. What's it going to take to get another couple of orders of magnitude? I don't know. 5 years, 10 years, who knows? Is it going to happen? Yeah, it'll happen. I don't know exactly, but we're moving in that direction. When I was alluding to the fact that we might see a 100-fold expansion, that was based on having received, one such machine last Sunday. We still got a lot to work at. If I'm giving you the sense of it's a rapidly moving world, we don't know exactly how fast it's moving, that's what's exciting about it. That's why we come in on Sunday, because the world is changing. It's different on Sunday than it was on Saturday. That's why young people come in and work weekends. Yes, maybe there was maybe one more question. So a last question, up in the back, yes. Yep.
How do you explain the large differences you see, physical differences between races?
The large physical differences you see. What large physical differences? Oh, maybe we're kind of tuned to really make a big deal out of the small physical differences that exist. On the whole, bones, organs, all this stuff, very common physiology. I mean, I suspect that we're highly sensitive to little physical differences that we observe. In addition, quite honestly, humans schlepped around the world. They went to different kinds of climates. There clearly were some genes that were selected upon for being in different climates, for resisting malaria and not being in malarial areas and all that. But we often imagine that those physical differences we see between people, which we consider as large physical differences, actually reflect a structure of variation where different ethnic groups are wildly different from each other. That's why I point out that 95% of the genetic variation is shared. 5% is local to different continental groups. 5% of the genetic variance is regional variations that might be found in Asia, Africa, et cetera, but it's a minority. When we talk about what causes prostate cancer or let's take prostate cancer. Prostate cancer is twice as frequent in African Americans as it is in European Americans. It's not, though, because African Americans have a different gene. It's probably because the same gene, in fact, we have a pretty good guess, chromosome 8p23 or something, the same gene is slightly more frequent in African Americans than it is in European Americans. those same genetic differences are all over the world, but they might be at slightly higher frequencies in some groups than other groups, et cetera. We can either choose to say, oh yes, well that's something that really affects African Americans, totally different, or we can just say, in fact, there's fluctuation in those frequencies of basically a common genetic heritage. So I kind of look at it and I say, fascinating differences. There are probably some natural selection involved for different environments. An awful lot of just random genetic drift as little populations went around the world. But when I look at the overall picture, I'm struck by how similar we are, how incredibly similar we are as a species and how much of our genetic variation is shared. I suppose you can look at the glass two different ways, but that's certainly my take on what I read in the Human Genome. The Human Genome is a great book to go to sleep with. You know, you open it up, it's long, it's really good, and you can open up any given night to a different page and read a different story. It's 1001, Arabian Nights. There are stories about evolution, there are stories about medicine, there are stories about physiology, there's stories about everything in it. We've only begun to read the book, but it's going to keep getting better and better. Hope you all come back next week and the week after and after that. Thanks for being part of the Broad.